So I'm going to take advantage of Burrett's excellent introduction to the area, and if you'll indulge me, I'm going to try and focus on just one experiment to give you an impression as to how we try and deal with some of these issues. So just a tiny bit of background for you. Of course, we know about the neurophysiology behind the bold signal. We know that we're not looking at neural activity. We know that we're looking at the consequence of neural activity, neurovascular coupling, hemodynamic effects, and also statistical effects as well that give us the outcome. And we know that in a voxel, really there's a lot of things going on. And the thing that's a minority contributor to a voxel population is actually the vasculature. So we're relying on 2% of a voxel to give us 100% of our signal in bold imaging. And of course, when you start to put drugs into the mix, it rather gets a bit complicated. So on the top left, what you can see is that the drug effects, of course, could affect the stimulus, which could have a subsequent effect on the neurovascular coupling and your bold signal. But of course, the drug could affect neurovascular coupling or the vasculature directly. And we also know that these changes, these potential changes, are very important. We've already heard how they could be important through these correlation analyses, but I'm just going to show you one picture from a particular study which shows this well, back in 2002. So that's on the bottom left. What you can see is a bold response, which are the little spikes in the grey bar. And if you alter the baseline blood flow, in this case with hypercapnia, which is in the red, you can see that these spikes are going down. So there's an interaction between the baseline state of the system in terms of the vascular response, measured with CBF here, and the actual task-induced bold response. So these are not theoretical concerns, these are real concerns. And this is from a paper by Richard Wise, which tries to summarize when you're doing a drug study, or of course if you've got a chronic change in a drug system, for example in a particular patient group, these concerns are equally valid. And the point of this is to show you in a similar way to the previous slide that you've got these different contributions to your bold signal, and while the red bars, your neural response, are what you want to see the consequence of if you have changes due to drug or being a particular patient in the neurovascular coupling or your vascular response, you might make an erroneous conclusion, which are the bottom two rows. So the sorts of questions we're asking at the moment, and we've been asking for a very long time, is how can we measure these main effects of drugs, these changes, of course, in the baseline? How can they relate to the drug targets? So we're measuring fMRI, of course. We're not measuring the binding of the drug in particular brain locations. So we're trying to sort of see if we can fill up this whole pathway. And how does that affect your bold response? Okay. And the study I'm going to show you and describe is a study looking at antipsychotic drugs, dopamine D2 receptor antagonists. And D2 is a rubric for the D2, D3 system. We know that this is a system where we should be very concerned about effects on neurovascular coupling for two main reasons. One, you've got changes in dopamine release when you give antipsychotics, and D1 receptors are present on the smooth muscle. And another reason, if you need more, is that D3 receptors sit on the astrocytes as well, so they can potentially affect the neurovascular coupling, the vascular response. So the data that was collected was a collaboration between our group and Roche Pharmaceuticals as well. And they were very kind in supporting this data collection. And what we wanted to do was understand the effects of different antipsychotic drugs. When you look at the comparison of antipsychotic drugs in clinical trials, they largely see much of a muchness. Okay, there might be some variation in side effects, there might be some subtle variation in negative effects with different classes, but they're largely similar in their effects. It's very difficult, of course, to do experimental studies in patients where the same patient will be given different drugs as well as a placebo. So we conducted this study in healthy volunteers so we could run the experimental protocol. We had 2 groups of 21 individuals, and one group was a dose-response study, which is risperidone was chosen because we could safely give risperidone at these 2 different doses that we chose. We chose a 2 milligram dose because the occupancy at D2 receptor just creeps into the clinically efficacious range, and we chose 0.5 milligrams because that's outside the clinically efficacious range. And then we did the same with haloperidol and olanzapine. Again, we're trying to choose doses that give us a clinically efficacious occupancy at the D2 receptor. And the volunteers did breath hold, they did blood flow scanning, and they did some cognitive tasks as well. I won't really talk in depth about the perfusion effects because they're published in the paper at the bottom by Peter Hawkins. And what we see is what we see in other studies, from studies we did with PET back in the noughties, studies we've done with ASL as well at 1.5 Tesla. But we see this very dramatic increase in striatal blood flow. We see some other cortical regions changing as well. But the striatal effects stand out, and they happen with all the drugs at all the doses. So the first result I'll show you is currently being presented in Paris by our student, Pierluigi. And what we've tried to do here is try to understand how these blood flow changes relate to the receptor distributions, first of all. Because, of course, this is blood flow, not any sort of pharmacological target imaging. And what Pierluigi did was he split the data up using an atlas, and using the same atlas, he aligned the data with a PET atlas for phallopride, which is giving you D2 receptor density across the entire brain. And also, I won't dwell on this here, but also we did the same thing with expression of D2 receptors, mRNA, from the Allen Brain Atlas. And then it was simply a cross-correlation across these maps that he looked at. And you can see here a very clear relationship, positive relationship between the blood flow response and the receptor density. And on the top, the striatum stands out on the top right, and even if you remove the striatum, these correlations remain. So that's kind of reassuring. And there's similar work done in the monkey by Christine Sander as well, building a model around the basal ganglia, but this is nice because it extends it into the human and it extends it out into the cortex as well. And you can see, do I have a pointer? Do you have a pointer? I'll point this side. You can see here that olanzapine here at 0.48, it has the lowest affinity for the D2 receptor, and it also has the lowest correlation as well with the blood flow changes. And with Jürgen Descartes, who's sitting in the audience, he took this data along with other groups and had a much closer look at these relationships with affinity, and that's published in scientific reports if you wanted to look at it. Okay, so what do we do about this with the fMRI? How do we take these blood flow changes and move them forward to help us with the fMRI? So we've added in the breath hold paradigm, so that's already been introduced to you. But very briefly, we want to measure the vascular response rather independent of task-related neuronally induced responses to the vasculature. Okay, so we see this as a putative measure of vascular reactivity. What's really nice with the breath hold response is that the signal variation does co-vary very tightly with the end-tidal CO2 measurement that you get as well. So it's a lovely validation of the work. And you can see in the bottom right from one of the many papers using breath hold tasks that the response to the breath hold is widespread across the entire brain, across the entire grey matter, and quite dramatic. And this is just from another paper here. I'll just point to it again here. You can see this is the bold response to finger tapping, and it's very, this makes the bold response look really rapid, like you've got high temporal resolution, but you haven't. And this is the breath hold response, which is much slower, peaking somewhere like 15 seconds later. And it's huge, of course, as well. And this is just one figure showing the effects of aging as well, which Bharat has already talked about. So it's very important, especially if you have a varied group, to take these sorts of things into account. So our breath hold paradigm was a breathe out and hold paradigm as well, and people repeated it across 5 blocks. So they breathe normally, they breathe out and hold, then they breathe normally, they breathe out and hold. So it's a block design, really. And this is showing the bellows response across the different drugs, and all I need to tell you is that there was no effect of the drugs. The drugs didn't make you breathe more deeply, they didn't make you breathe more shallow, they didn't make you breathe in earlier or hold your breath for a shorter or longer time. And I think it's very important that you take these into account when you're doing these sorts of paradigms. And this is the response, this is the difference in the breath hold response between the drugs. And I think the thing that struck us the most is that there was a difference between these drugs. So of course, ostensibly, these drugs are in the same class. There are some variations, but it was olanzapine that caused a big reduction in the breath hold response, again, across most of the brain, which you can see in the blue on the brain, overlaid on the structural image there. So haloperidol and risperidone, both doses had no effect. So then you've got one drug having a big effect on breath hold. What I didn't mention before is that olanzapine was the drug that didn't really have a main effect on the blood flow as well. So you've got this dissociation now. You've got drugs like risperidone and haloperidol affecting blood flow very strongly, absolute blood flow with CBF. And then you've got olanzapine, which is affecting breath hold, but not blood flow. So unfortunately, taking just one of these measures wasn't enough. We use the MID task. I'll just show you the data from one task and one analysis so I can finish on time. The MID task will hopefully be familiar to many people. It's a task that's trying to get at elements of reward processing, reward anticipation, reward receipt in particular, and you have a target you have to respond to quickly. If you respond quickly, you get a reward, and it's probabilistic as well. And we get nice activations across areas that you'd expect, including the striatum and the ventral striatum. So I'll just focus on an ROI analysis for the remaining bit in the ventral striatum. And again, we see a difference between the drugs on this task, where risperidone at the clinically efficacious dose has an effect on the ventral striatal activation with this task, whereas actually the lower dose does not have a significant effect. So this is unlike blood flow, where both drugs had effect, so we couldn't really discriminate between them using multivariate analyses. And then with haloperidol and olanzapine, again, we saw a difference with one of the drugs, but not the other. So olanzapine had an effect, but not haloperidol. And the important bit is rather excessively written only in words, which is that when you control for the blood flow and also the breath hold, what we find is that the results do change, but the critical result here in the ventral striatum remains significant. There are other results, if you look at the whole brain analysis, which contract, and many of them go away, in fact. So there's lots of results that occur with the drug, but the predicted results in the ventral striatum rather hold on to these controls and these confounds, whereas other results wax and wane. So it's been very important in trying to help focus our conclusions around the ventral striatum, because these analyses allow us to do so, whereas if we just took the bulb by itself, we'd find ourselves getting lost down some alleyways talking about some other regions we weren't sure about. Okay, so just to summarise, there's no summary. To summarise what I summarised just now, just to thank people. So Pierluigi and Peter have done the analyses that I showed you. Peter collected the data. Indaba and Steph are our medical team, and Mattia is our collaborator. And thanks to the Biomedical Research Centre and Roche for funding.